Hello again. I will present you now the project on the emergence of tuberculosis at the human-elephant interface, which will be conducted in collaboration with different institutions in Laos, the Centre d'Infectiologie Christophe Mérieux, the Lao Tropical Institute, the University of Health Sciences, the Elephant Conservation Center, and the Institut Pasteur in Cambodia. Over 10 million people worldwide are infected with tuberculosis today. In Laos, TB and drug-resistant TB represent a significant public health threat. The disease is also threatening elephants and other animals like sun bears. Asian elephants are victims of shrinking habitat, poaching, human conflicts and diseases, and are listed as endangered on the IUCN Red List. Captive elephants are employed for tourism, festival purposes, although hundreds still work in illegal logging. In Laos, owning an elephant is still a conventional practice. The constant proximity between the elephant and the mahout, who is someone who owns and works with an elephant, increases the chances of diseases transmissions in both directions, notably TB. This has already been documented among keepers and elephants in zoos and sanctuaries across the world. Mycobacterial culture from collected sputum, accompanied by chest X-rays, is still the gold standard for diagnosis of active TB in human beings. For the diagnosis in elephants, veterinarians are required to collect trunk washes or blood samples from trained animals, with results of varying degrees of sensitivity and specificity. These techniques are not applicable to untrained elephants, and therefore a diagnostic method based on non-invasively collected samples would be a much more suitable approach. Alternatives exist today to detect TB in fecal samples from people, and notably sun bears, with active disease. Evidence shows that swallowed Mycobacterium tuberculosis may survive the enteric tract, suggesting that stools could be used as an additional or alternative specimen for the diagnosis of pulmonary tuberculosis. This non-invasive procedure is of particular interest for patients who cannot expectorate, like immunocompromised patients and children, and for elephants and other animals for which other biological materials would be stressful or difficult to collect. The main objective of this project is to determine Mycobacterium tuberculosis prevalence and antibiotic resistance among captive elephants and their mahouts to better evaluate the risk of TB emergence at the human-animal interface in Laos. More specifically, we aim to develop a reliable, more sensitive and specific test to detect Mycobacterium tuberculosis and antibiotic resistance in non-invasively collected elephant fecal samples, based on the techniques and knowledge acquired by our Cambodian partners in TB diagnosis in sun bears. Determine TB prevalence and molecular characteristic of this pathogen infecting captive elephants, the mahouts and their closest contacts in the household, and address how TB prevalence and Mycobacterium tuberculosis molecular characteristics and antibiotic resistance are epidemiologically linked among the aforementioned subjects. Elephant fecal samples will be collected in Dongmitsai district and at the Elephant Conservation Center in Saeburi district and will be sent to the Institut Pasteur du Cambodge for analysis. Sputum and or fecal samples for each mahout with suspected TB infection will be collected and sent to the National Reference Laboratory for TB in Vientiane, the CLM, for culture, molecular identification, and drug susceptibility testing. Thanks to the expertise acquired on the detection of TB in sun bears' fecal samples, we will adjust, improve, and validate the techniques of TB detection in elephant fecal samples. Nucleic acid-based amplification tests will be carried out to detect the pathogen in human and elephant stools by using the GeneXpert diagnostic test, allowing for the detection of the Mycobacterium and the rifampicin resistance, by extracting DNA, conducting real-time PCR amplifications and detection of IS6110 or RPBO POB genes, followed by DNA sequencing and phylogenetic analysis. Standard PCR and sequencing reactions will be performed for positive cases in order to confirm the specificity of the real-time PCR results. From February to March 2022, We will focus on field preparation and administrative authorization requests. In April, we will conduct a prospective tour in Tongmitsai district to select the Maout families, followed by a preliminary information mission and participation signed agreement. We will then collect sputum and/or fecal samples in human beings and captive elephants, and from June until the end of October 2022, we are going to conduct the laboratory work and analysis in Vientiane for the human samples and in Phnom Penh for the elephant samples. And the month of November and December 2022 will be dedicated to the scientific valorization. So this pilot project has been conceived also with the idea of extending in the future the sampling to the existing 450 domesticated elephants working in Laos, and also to their wild counterparts because of the interaction between the domesticated females and the wild males during the mating season. Thank you again for your attention.